Hope everyone is doing well today. We'll be getting started with the webinar shortly. All right, we're going to go ahead and get things started. Greetings all. I hope everyone is doing well today. My name is Christopher Walker, Senior Program Analyst at the National Environmental Health Association, and we are happy to be hosting today's Sterile Insect Technique webinar, which is in promotion of this week being National Mosquito Control Awareness Week. We are happy that you are here today to learn more about this technique. There will be a Q&A period after the conclusion of the presentation, so please make sure that you ask questions in the chat option. Also, at the conclusion of this webinar, please complete the survey that will follow once you hit end. So we look forward to your responses on that. This webinar is hosted and promoted by NEHA's Vector Program Committee, which the committee is comprised of local, state, and federal and industry professionals with experience with vectors. Our mission is to be a resource for environmental health professionals and NEHA leadership dedicated to developing resources, programs, and fostering improved vector control programs. And now I would like to introduce our committee chair, which is Dr. Brooks Bose, who's going to be moderating this webinar. Dr. Brooks Bose is the Director of Research and Innovation for Vector Disease Control International and has a PhD from Colorado State University. As a specialist in emergency response, she has developed protocols and coordinated surveillance and management operations with various local, county, state, and federal organizations. In 2016, she served as the on-site coordinator to both the Virgin Islands and Marshall Islands after the emergence of Zika virus, and served as the IPM manager for VDCI's hurricane response efforts, more than she cares to say. She is the industry director for the American Mosquito Control Association, is chair of our Vector Program Committee, and we are happy to have her here today to moderate. So Dr. Brooks Bose, I hand things over to you. Welcome.
All right, well, thank you, Chris. As Chris said, this is the National Mosquito Control Awareness Week, which is sponsored by the American Mosquito Control Association. And it was initiated as an effort to remind the public about things they can do to help protect themselves from mosquitoes and the diseases they carry. It also serves as a platform for educating the public about the tools used for reducing mosquito populations and how an integrated pest management program works. So the Vector Program Committee is really excited to have two amazing speakers from districts within the state of Florida who are implementing multiple kinds of sterile insect technique strategies. So next slide. So for a little bit of history on mosquitoes, for those of you who don't necessarily work with them every day, it is only the females that bite. So a lot of the sterile insect techniques are targeting only the male mosquitoes through various types of strategies. The males are modified so that they do not have reproductive young or that they do not make it to adulthood in some form or fashion. Because the males don't bite, they are not considered a vector of public health importance. Only the females are. And the release of the altered males does not have any kind of non-target effects given the strategies that are used. Next slide. So there are three basic strategies, or three strategies that are currently being explored for sterile insect technique in the US. The first is X-ray irradiation to sterilize the male mosquitoes, which are then released to the wild and mate with females and produce non-viable offspring. There's naturally occurring bacteria, which are introduced and create some kind of incompatibility between the males and the females. Or there's genetic modification through gene drive technology. So our wonderful speakers today will be talking about the first and third ones on there. And so our first speaker is Andrea Leal from the Florida Keys Mosquito Control District. She has a bachelor's in biology and marine science from the University of Florida, has a master's in entomology from the University of Nebraska, and is former president of the Florida Mosquito Control Association and involved on numerous panels and activities within the industry. So with that, I'll turn it over to her and look forward to hearing what she has to say.
Okay, thank you, Brooks. And I really appreciate the opportunity to be here and talk a little bit about our Oxitec genetically modified male mosquito releases that have been happening these last couple of years. Next slide, please. A little bit about the Florida Keys Mosquito Control District. We are located on the southernmost tip of Florida and cover all of Key West to Key Largo. So it's a very long island chain. Of those islands, we can probably treat maybe 70 or 80% of those only with naturally occurring products because they are protected lands. So we are in the middle of a national marine sanctuary as well as full of other endangered species and things like that. So, we like to try to be on the cusp of new technologies to make sure we're doing the best for our environment. Our district has about a $15 million operating budget. We've got about 70 full-time employees and 6 aircraft. So we are a very robust mosquito control district. Bit of feedback. Okay. As far as mosquitoes in the Florida Keys go, as you can see here, our main disease vector is Aedes aegypti, and that's what a lot of these sterile insect techniques are targeting. And Aedes aegypti only makes up about 2% of our collections that we have throughout the entire Florida Keys. So very, very small percent of our mosquito population, but takes up over 10% of our budget and actually takes up about 50% of our chemical budget. So as you can see, it's extremely expensive and very difficult to control just that minor population. Next slide, please. So how we kind of started all of this in Florida, a lot of us were built on nuisance mosquito control. However, over the last few decades, we've seen more and more introductions of mosquito-borne diseases. And then for us in South Florida, we started seeing locally acquired cases, specifically of dengue fever, chikungunya, and Zika virus, starting with us in Key West in 2009 and 10. And then you can see from the chart there that we've had recurring locally acquired cases, specifically of dengue, almost every single year in the state of Florida. Next slide. And then probably a couple clicks. Yeah, and one more. There we go. So this indicates really a map of where we've seen locally acquired cases of dengue fever. In 2009 and 2010, we had our first outbreak of dengue fever in the Florida Keys since the 1930s, where we saw about 95 cases down on the southernmost tip there over Key West. We threw everything we had at it, added more personnel, and really didn't see anything until we fast forward to 2020, where we saw just over 70 cases of dengue fever on the opposite end, 120 miles away in Key Largo. So, we really were focused very hard as far as disease vector control in Key West because of that outbreak. However, I think what we've shown now since 2020 is we really could see these outbreaks taking hold pretty much in any areas where we're seeing Aedes aegypti, especially with all of the travel that we see throughout the Florida Keys. Next slide, thanks. So as far as our current Aedes aegypti control measures, Aedes aegypti is one of the most difficult mosquitoes to control. They breed in any little tiny bit of standing water. So it takes a lot of people and a lot of time to go around house to house, door to door, and find every little bit of water. So what we've incorporated, as well as going house to house, is also wide area larviciding, which is basically a treatment of BTI by air that we apply as a mist over residential areas. We've seen a good control with that. However, as we've shown, just because we're getting a good control doesn't mean that we are, below disease threshold. And another thing we're contending with is product resistance. So adding all of that together, the difficulty to control these mosquitoes, the diseases that they're responsible for transmitting, and the resistance issues that we're seeing with some of our adulticides, we've really shown that mosquito control needs to continue looking for new tools to integrate into all of the tools that we currently have to try to figure out the best way to control the Aedes aegypti species. So back in 2010, we started discussions with Oxitec, and at that time, Oxitec had just done some work in the Cayman Islands. So we were looking at the potential for use of genetically modified male mosquito releases to help us with our Aedes aegypti. However, in 2010, we had no idea how to do that. The regulatory wise. So it took us a good 4 or 5 years to even have a federal authority say, yes, we will regulate this. And so 2015, FDA took oversight. We did a lot of work locally to try to tell our people what was going on, what we were looking at doing, and actually did a local voter referendum because there was a lot of question on, what is the acceptability of genetically modified mosquitoes? So on that local voter referendum, we asked, registered voters, do you support moving forward with the release of genetically modified male mosquitoes? And as you can see there, 31 out of 33 voting precincts voted in favor of it. So that kind of gave us the green light to continue pursuing this. However, again, federal oversight switches now from FDA to EPA, and it took us another 3 years to get all of the approvals in place that we needed on the federal, state, and local level. So as you can see, it's been kind of a winding regulatory pathway here, but excited that after a decade of work, we were finally able to get the project started. Next slide. So as you guys can imagine, one of the biggest issues, of course, is communication. Trying to get people to understand what we're doing and why we're doing it and how we're doing it. So of course, genetically modified often has a perception of being negative. So, we really had to take the time to explain to people, most genetically modified organisms or foods that we know of are to make something more robust. Specifically in agriculture. However, this genetic modification is the opposite, right? So we're releasing males that will mate with wild females, and then all of their female offspring will die. So it's really a big difference, and trying to get that information out to people has been difficult. And as you can see here are a couple of examples. There are some vocal minorities in the Florida Keys that rented billboards to tell people, say no, or, you're going to get a bite reaction. So it's been a battle for sure, trying to tell people, what this is and why we're doing it. So the first thing we did before we did any releases was to do a communication survey with our residents. What do you know about the project? How do you get your information so we could make sure that we're pushing information in a way that most people are going to receive? And we really focused on our message. We do weekly radio spots still to this day. We have printed mail materials, and we made a dedicated project website. So it's kind of a one-stop shop for anybody that you could join the project, ask to be a host for a release box or a trap. You can ask for one of our staff to come and give a presentation. So it really is a great tool to use. But, number one is to know how best to reach people out there. One of the best tools I thought we did were monthly webinars, and we've actually started those up again. We did 15 monthly webinars that began in July, every different topic that you could think of under the sun. And our biggest thing that we wanted to do is make sure we're answering everybody's questions. So we have about a 30-minute presentation and then 30 minutes for any questions at all from the public. So we began releases last year, and those were very small pilot projects, both a single point release project and a multi-point release project. And what we were really trying to do is really just get those building blocks as far as, how far are those males flying and how long do they last? Are they utilizing natural breeding sites? Are we seeing what percent kill of female mosquitoes are we seeing? And then just making sure that there was a good mating ratio between those males and our wild females. All of the data that we collected were used, we used egg collection ovitraps, adult mosquito traps as well. And then a nice method to really see what was going on is that those Oxitec males have a fluorescent gene. So you can look at all of those field collected larvae and adults, and if they fluoresce under those microscopes, it is, it's genetically marked to know if it is an Oxitec mosquito. So I'll kind of go over some of the things we learned this year. The flight radius was confirmed, and you can kind of see the layout of the two different trials with the single point on the left and then the multi-point throughout a neighborhood on the right. And so we did find that the flight range of Oxitec's male mosquitoes is basically the same as those wild Aedes aegypti males. Another thing that was confirmed, because you do get a lot of questions when you tell people male only releases, because it is genetically selected for males only. So we did also prove that no females were released at all, and everything that came out of those release boxes were male. Next slide. So another point was that the larvicidal efficacy of those Oxitec mosquitoes were 100% throughout the project. So if those males were breeding with wild females, all of those fluorescent larvae that were collected, we showed that only the males survived. And that was in all fluorescent collections throughout both trials. Excuse me. Okay, next slide, please. Additionally, those mosquito larvae were found in a variety of different breeding sites. And you can see some of them listed here: flower pots, plant trivets, garbage can lids, buckets, tires. So we were excited to see that, while the males were mating with our wild females, those wild females were spreading those progeny throughout sites and different breeding areas. So those were really the building blocks that we learned from last year's trial. One of the things we did not really get a lot of information on is really the overall adult population suppression. And that was because we had 2 tropical storms in the middle of the field trial. Field trials never go how anybody wants them to go. So there were big
Andrea, are you still there?
control mosquitoes around that one household, and then also a marker capture study. So I think we're in a really great spot to continue gathering data, and hopefully, in the very near future, we can talk again about, how well the population is actually suppressed. And just thank you for your time. I really appreciate being here.
All right, thank you, Andrea. So our next speaker is Rachel Moriali from the Lee County Mosquito Abatement District near Fort Myers, Florida. Rachel earned her undergraduate degree from the University of North Florida and a master's in medical entomology from Georgia Southern University. She's been working on the development of Lee County's sterile insect control program since its inception in 2017, and we are fortunate to have her here today. So thank you, Rachel.
Thank you, Brooks. So thank you for having me. Again, this is another Florida program for you. And I will begin then. Next slide. Okay, so if you're not familiar with Lee County, we're down in Southwest Florida and just a little bit north of the Keys. We've got a ton of salt marsh habitat similar to what Keys has to deal with. So historically, a lot of our focus has been on the more pestiferous Aedes tenurinus. However, this is not the focus of the program that I'm talking about today. So we have a very comprehensive mosquito management program. We've got larviciding capabilities and adulticiding along with ground and aerial We're a very robust program up here. So, next. So we went into what sterile insect technique is a little bit, and that really does focus on the release of sterile organisms for the population suppression. Traditionally, it focuses on the release of males. And the type that we're using in Lee County is classical SIT. So it's kind of the, I guess, more basic one. We're using irradiation to induce the sterility of our males. We began this program back in 2017, and that really was foundational with a meeting with the International Atomic Energy Agency. They provided us a lot of insight in the ways that we should be setting up our program and things that we should be focused on as we moved forward. In doing so, we decided that we would be focused on Captiva Island as our area of interest. And that was for a couple of reasons. One being that it had an isolated population, which we could access by road, but it was kind of far away from the mainland. We wouldn't have immigration between the release site and the rest of the mainland. And we have resistance issues like almost everyone in the world with our Aedes aegypti. Next. We had some town hall meetings. We met with the Captiva Chamber of Commerce. We met with the biologists out there. We have local media coverage. This was all to really get the public on board with what we were doing and keeping them abreast of what developments we had. We had them out there for our first mark release recapture. They got to actually see, as you can see in one of the images, they got to see the mosquitoes coming out. And it was a really good opportunity for them to be involved as well as us. And they were actually out there for one of our first releases operationally as well. So we really enjoy our relationship with our public media to be able to show what we're doing for the county. So for doing these releases, you really have to know what your population is doing. So we have a very in-depth field surveillance program, and that was all to collect this entomological baseline. We began surveillance of our mosquitoes on Captiva in 2017, and this was through BG Sentinel-2 traps and oviposition cups. So every site had a trap and a cup, and the traps were set twice per week. The cups were maintained once per week. This provided a really in-depth view of what the activities were at the moment. You got to see what the adults were doing, and you got to see how the eggs would change over time. We divided our island up into 3 different areas, what was known as a treatment zone, a buffer zone, which was the last kilometer of Captiva Island, and a control zone. The control is located on Sanibel Island, and that's an island just immediately south of Captiva Island. It still has the same characteristics of Captiva, so it was a good spot for us. So this is what it looked like. That's a whole lot of pins on one little island. And our traps are set every 200 meters. So this is actually a pretty large program. There are 45 traps in the treatment area, 3 in the buffer, and 10 in the control. And you can see the control is the sea down on Sanibel Island. So this gave us a really in-depth view of what we were working with out there on the islands. We were able to see our seasonal fluctuations over time and how our population behaved seasonally. We actually got to see what the impacts were due to hurricanes, as well as we in 2017 had Hurricane Irma bust through the island. And in 2018, we had a bit of a lower population than expected due to the hurricane effects of the previous year. But yeah. So as this was all going on, we were working on the lab space that we would be using to rear our mosquitoes. So we ended up using a building that was already existing at the district, but we were doing building modifications to it. So the building had normal access doors. We changed those into no outside entry from them. You could only use them as exits, except for one door, which was the point of entry. And then that entry point was protected with an air curtain, and we have a PVC strip curtain as well to try to prevent any mosquitoes from exiting. We replaced any of our drop panel ceilings so that the sheetrock panels would be more humidity resistant, because hey, mosquitoes love humidity. And so we also got some new humidifier systems so that they would be supported in these environments. We got some very special lights as well. They mimic sunrise and sunset because they turn very slowly on and very slowly off. And that really helps set what's called a photo period for the mosquitoes. So when they are released out in the field, they're used to what the field conditions would be. We do a lot of mosquito production, and all of our operation is in-house. So all of the work that we do is done by myself and my team. We do our larval rearing in these closely stacked trays you can see on the top. These were produced by Wolbachia Biotech, and they're modified off of an International Atomic Energy Agency design. But they can hold, they have 40 trays per rack, or per side, and 2 sides per rack. So it's a total of 80 trays per system, and we have 6 of these. We feed them fish food throughout their larval stages, and then once they pupate, we drain them off, and then we perform pupal separation. And that's how we're actually able to get our males separated from our females. They have sexual dimorphism, so the females are larger than the males, and then we can collect them separately. So if you want to play this video right now. Nope, wrong one. Okay, well, nope, okay. So anyway, the automated pupil separator over on the right-hand side is how we separate the males from the females. The females, because they are larger, will get caught higher between the glass plates, and then the males get caught at a lower level and the larvae pass through. So it's a very efficient system and has allowed us to really increase our production. We also have to maintain we have to maintain our adult colony as well. We have lots of cages going all at once to make sure that we're collecting plenty of eggs. You have to have the eggs to support the program. And our cages are stocked at 3 females per every male. And that actually reduces a lot of the male harassment issues that we can face, so that we can actually have even better egg production. We give them defibrinated sheep blood for eggs and 10% sucrose for energy. So the way that we induce our sterility, again, is by X-ray. It is ionizing radiation, which if you look at this video, that metal point in the center is where the X-rays are emitted from. It's kind of like a cloud around that center point. And then the canisters rotate around. I like to say, like a really not fun Ferris wheel for them, but that helps to ensure that they achieve an even dose of the radiation as they move around that center point. And we're currently working to switch to adult irradiation, but that is not part of the program at the moment. And we're using 42 gray to achieve this sterility. My crew consists of 3 assistants. Those are our field personnel. They do the trapping. They help with assorted tasks that are needed. We have 3 different technicians. They're in the lab actually doing the daily mosquito production of mosquitoes and performing some colony maintenance. There is Stephen, he is our supervisor, and he is in control of the daily production and the field needs, overseeing both the assistants and the technicians. We have a quality control specialist. She focuses on making sure that we're achieving the parameters that we've set to have quality, healthy mosquitoes that are going out into the field. And then I am the one in charge of it all and making sure everything goes right, hopefully. So we've also performed a mark release recapture, actually several of them. And these have looked at our flight range, the dispersal of the mosquitoes, and how long the mosquitoes survive out in the field. They also help provide us population size estimations as well. For these, we operated 40 BG traps, and they were maintained every day for the duration of the study until we stopped collecting any more marked males. That's quite a bit of effort to keep doing that, but it provided us really good information. And so this consisted of both single point and multiple point releases. They each provided good information, but different information. And they were conducted throughout the season, so we could see what the population would do at those points in time. This is a manuscript that is submitted, and we're waiting for the reviewers on that. So operationally, this is what we're doing now. We are again irradiating our pupae at 42 gray. Then they're marked with fluorescent powder. Ours don't carry a gene for that. We just apply a powder. And then we can see the powder when we recapture them under UV light. So they're sugar fed until they're released, so they have all the energy that they possibly can. This was begun on June 10th, 2020, and has been continuing with usually 2 releases per week, every week since. And we have released, now this year, it's up to over 15 million, I believe. So our releases have been increasing in time, and we're being more productive with our mosquitoes. And again, always quality control is assessed on our mosquitoes to make sure that they're healthy. We're working at, it's about a 600,000 per week, 600,000 sterile males per week. And then we're working to increase to 1 million, hopefully soon. So this is really the exciting stuff. What have we actually seen? Our releases began, as you can tell by that red line, in 2020. And if you look at the orange line, that's our control area on Sanibel. It went through its normal population fluctuations over the season and had a very, very productive 20. On the other hand, our blue area represents our release area on Captiva. That population was actually having a very good year before our releases began, but then once they started, the population effectively took a nosedive, and then it just totally failed to recover. What we were seeing was reflective of winter numbers during our summer period. And this is also reflective. This is a heat map basically saying the same thing. You can see visually where the proportion of the adult Aegypti were caught. And then once you start seeing the releases occurring in 2020, you see a lot more blue than black, indicating absence. So the population in 2021 really showed the impacts that sterile insect technique had on the Aedes aegypti out there. So just to kind of sum things up, this is a really safe and environmentally friendly technique. The mosquitoes that we're using are local, they're our own, we colonize them ourselves, and they're produced in our local lab. They're species specific, so we're only working with Aegypti here. No other species are going to be impacted from this. And resistance that we face in the field, because this is just the males mating with the females, and it can be effective. So as we've demonstrated with our population, we were able to achieve suppression with it. There are challenges though. This is not the answer for everyone. There is a major economy of scale question at hand. It right now is fairly expensive to do until we get better technology to really support what we need. I would not say this is necessarily a feasible thing for everyone at the moment. Hopefully in time, it becomes more accessible. One of the reasons is because it is labor intensive. My team is so hardworking, but it's just a lot of work to rear these mosquitoes and get them out there and making sure that the surveillance is constantly happening. So it's an emerging area. I am happy to keep talking about it because I think it is so promising. But it's if you're interested, you can reach out to the International Atomic Energy Agency. They have a lot of great publications on their website. And for us, in contrast to what they've had to face in the Keys with the GM mosquitoes, we don't have to have special permitting for these mosquitoes. We can actually, we haven't had any regulatory hurdles at all. And yeah, that's pretty much it. I'd like to thank my team, they're the best. And Erin, our assistant director, Dr. Cole, our executive director, our visiting expert who is now currently sitting in the lab is Danilo from IAEA. He's been a profound resource for us. And we were supported by a grant through the Florida Department of Health and CDC as well. Thank you.
All right, well, wonderful. Thank you to you both. Those were great presentations and really interesting stuff that we have going on in the industry. So if anybody has questions, please submit them in the chat and our speakers will be around for a little bit to answer. So I had a question about what type of traps are you using when you're doing the invasive Aedes surveillance? Are you using BG Sentinels or what type of traps is the best one?
We use BG Sentinel 2 traps in conjunction with oviposition cups. So I highly encourage the BG Sentinel 2s.
Yep, same on this end. Very versatile, easy to use, and do a great job in collecting Aedes aegypti.
And do you use ovicups as well, Andrea?
Yes, yeah, throughout all of the treatment and control areas.
Wonderful, thank you. So it looks like there's a question for Rachel. Why is switching from pupa to adults beneficial?
Great question. That will actually help a lot with the timing. So in order to achieve the sterility that you need, you have to actually radiate at a proper, so that's 30 to 36 hours of pupation. That is sometimes very tricky. If your batch is not behaving the way that you hope, then you're basically chasing the time with them to make sure that you're irradiating at the proper time. If you radiate them too early, you can induce mortality. If you radiate too late, you don't achieve the sterility that you hope for. So having that really focused time period for the pupae can be a challenge. On the other hand, with the adults, once they emerge as adults and have a chance to basically sit there for a minute, they're fine. You can irradiate and have a lot more flexibility on their age. You're not so constricted. So it's really more about the timing and making sure my crew doesn't go crazy and have to come in at odd hours of the night.
Great, thank you. We have another question. Are there any environmental concerns when looking at the elimination of a mosquito species? For either one of you.
This is an invasive species, so it's not native here. We're not concerned about this at all because it wasn't supposed to be here in the first place. So no ecological impacts from that.
Yeah, additionally, I think it's important to know that these mosquito larvae are found in man-made containers. So there aren't natural predators that you find alongside them. And the population size is such a minute portion compared to naturally, mosquitoes that did come here or are from here. So I don't think that you're ever going to see any sort of impact other than reducing diseases, which, is good for all of us.
Excellent. Andrea, have you found this technique to be successful in reducing your chemical budget in the areas where it's treating? You mentioned that 50% of your chemical budget is focused on this species. I'd be curious how you see this changing that.
So because there is no known cost associated with the Oxitec mosquito right now, so we're basically operating under an experimental use permit. The product isn't registered, so we have no idea what the end cost is going to be. So ideally, would it be great to say yes, it would, of course. However, realistically, I can't say that yet. But like any other product, once something becomes registered, we'll do a cost benefit analysis and hope that it fits into our program in the right way.
Wonderful. Well, I don't see any other questions on here. So if there are any more, please type them on the chat. We'll hang on for a minute or so. Otherwise, thank you to everyone for attending, and thank you to our speakers for taking the time out of your busy day. I know that this time of year gets to be really busy with mosquitoes across the country. It's hot and humid, and so remember to wear your repellent and stay safe out there.
Awesome. Thank you, Brooks, for moderating. Thank you, Rachel, and thank you, Andrea. Great presentations, definitely informative, and you definitely see the opportunities as well as the challenges of this type of technique. So thank you all for being here. We thank you for the attendees for listening in on the webinar. If there are no other questions that I see, we're going to end things. We hope you have a great day. Thank you all for attending, and please make sure that you respond to the survey at the conclusion of this webinar. So thank you all very much again, and have a great rest of your day. Well, before we leave, I see one more question. I don't know if this one came up, but what are benefits of using GMO and or sterile technique over using Wolbachia method? Has that one been answered?
I can kind of start, and I'm sure Rachel can jump in. We've actually done trials with both the Wolbachia mosquitoes as well as genetically modified mosquitoes. There is a bit of a difference because with Wolbachia, you're releasing adult males, similar to what Rachel's doing there in Lee County. The Oxitec mosquitoes, we're actually putting eggs in the field and then waiting for those eggs to develop, and then the males kind of release themselves. So logistically, it's a little easier, but at the end of the day, I don't think we have enough information as far as efficacy goes. And we've seen pretty comparable efficacy in the publications so far. So I don't think Wolbachia is off the table for anybody either. I know, they had great results using that in California. So I think at the end of the day, when it comes to all of these techniques, we all just kind of have to look at, what do we have available? What areas do we have available? I know, I could never build a lab like Lee County has here in the Florida Keys, which I would love to, but, we're limited with space. So there are a lot of different factors that go into things. And I think it's important that anyone looking at any of these techniques takes all of those factors into consideration, because we are seeing great efficacy with all three.
Yeah, I'll second that. I think it's really about what's appropriate for you and your district. There's not any one right answer. These are all of them are emerging technologies and techniques. So I think they're all at this point pretty viable. And with the sterile insect technique, I know that it's very safe. If we did release a female, she's going to die very quickly because the X-rays actually impact her more than a male. So there is a challenge with Wolbachia. You got to make sure that you do not release any females there, because if a female does population replacement, then you don't achieve the cytoplasmic incompatibility. That's what's required for the population suppression. So that's an entirely different Wolbachia technique though. So I just say that they're all really viable at the moment. It's just what's appropriate and what your public will accept.
Yeah, I'll just add an interesting fact on Wolbachia. So in the US, Wolbachia is introduced to the male populations, which make them incompatible with the females. But in Australia and other parts of the world, they're introducing Wolbachia to the females, which actually reduces their vector competence and makes them less likely to transmit a disease. So interesting that there's multiple ways that can be used, but Rachel's absolutely correct. If it gets into both the males and the females here, then that would reduce the impact of the cytoplasmic incompatibility. So interesting, cool stuff.
Awesome. Thank you all. That question just came up and I figured, hey, why not ask it? So thank you all again for a wonderful presentation. Thank you all for attending. And I think now we are done for the rest of this webinar. So thank you all very much and have a great rest of your day.
Yep, you too. Bye.